Okay. So this is dealing now with not just one gene, but two genes. We talked a little bit about the law of independent assortment yesterday, and the point of that is that because of independent assortment, we can get any of this allele and any of these alleles in the same gamete, right? So talk about how you could physically draw the FOIL lines out so you could see all the possible combinations. In this case, parent one is really only going to give two different possible combinations in their gametes, right? They're either going to give a big A and a little b, or a little a and a little b, right? Parent two, do the same thing. This A and this B, that A and that B, that A and that B, that A and that B. They're also only going to give two possible combinations, right? Either big A, big B, or little a, big B. Everybody with me so far? Okay. So when you set up the Punnett square for this, do you need to make 16 squares? No, you only need to make 4. You only need a square for this, a square for this on one side, and a square for this, and a square for this on the other side. Those are the only possible things we can put together. Okay? So that's what you're going to do. So do that real quick. You can get the answer anyway. We're going to wow, that's like a trapezoid, not a square.
So could we end up doing like a Punnett square that's literally like a 2x4?
Well, generally it's an even number of possibilities because every gene has like potentially either 1 or 2 possibilities.
Oh, sorry. So like a 2x3.
So you could have a 2x4. If we did 3 traits, then you're going to have like potentially an 8x8 or a 2x8 or 4x8. But they're going to go by powers of 2 generally. So when you do this square, again, because still making only 2 possible combinations, you can make a smaller square. We end up getting 3 possible genotypes. How many possible phenotypes? 2, yeah, because both of these genotypes are going to show both dominant traits, right? So 3 out of the 4 are going to show both dominant traits. And 1 out of the 4 is going to show a recessive trait and then the dominant trait. Right? So this looks more like a monohybrid cross. We're just dealing with 2 traits. Okay, so that's Sometimes if you really like this Punnett square method, you can use this for dihybrid traits. We are going to talk about a second method that I prefer personally over doing Punnett squares for more than one gene. But before we do that, I want to hand back your quizzes. And I want to take a look at that meiosis diagram one more time. So most of us did really well on the quiz and on that diagram, but it ties in really nicely to what we're talking about today. So one more time. So you want to call your name if you can come up and get your quiz, and then I'm also just going to give you one more blank template of this. We'll walk through it one more time together. Lexi, Dylan, Sky, Jack, John, Kat, Aaliyah. Oh, sorry, Aaliyah. Aaliyah. Kara Burks. Cooper. Will. Julie. Conquest. You're welcome. Leah. Jada. Quiz. Maxwell. You're welcome. Jacob. Alex. Matea. Hannah. Carter. And Brayden.
Thank you.
So take a look at those comments there. The table had the most issues with the second and the fourth row. So ploidy level, that's dealing with whether the cells are diploid or haploid, right? How many sets of chromosomes they have. So for mitosis, you're going from a 2N to a 2N, diploid to diploid. Meiosis, you're going from diploid to haploid, 2N to 1N. The fourth column, I think, is maybe the most, or fourth row, maybe one of the most important rows in that column, in that table. Mitosis is going to make cells that are genetically identical. Meiosis, the daughter cells are going to have exactly half. So one of each homologous chromosome from the parent cell is going to go into that gamete. And those gametes are all going to be unique from each other. So one kind of, I don't know, it's really hard to explain with people were saying like half from mom and half from dad. That is true in fertilization. So when you go through fertilization, you're taking two haploid cells and you're putting it together. So it's not till fertilization that you're getting half of moms and half of dads. Okay. Meiosis, you're mixing up the alleles of the parent's parent. Okay, the paternal and maternal alleles are getting mixed up, but you're not necessarily getting exactly half of your maternal and half of your paternal chromosomes. You're going to get different ratios of those. So I hope that you can see that clarification there. I put notes on a couple people's quizzes about that. Number 2, What I was really looking for is a description of either crossing over, independent assortment, or random fertilization. You couldn't just list the names. You had to tell me what happened. I did also end up taking some answers that had to deal with our chromosome abnormalities. So some people mentioned like deletions and duplications and stuff, and that does increase genetic variation. A lot of times not in a good way, but so I took that answer also. Number 3, almost 100% on that. I and J, you take a look there. I is specifically denoting E, F, G, and H. So I is really just pointing to mitosis. Okay. If you put mitosis I and cytokinesis J together, then you get the M phase. Okay, so the cell division portion of the cell cycle, the M phase, includes both mitosis and cytokinesis. So I didn't knock off any points for that, but I did clarify that on your quiz. Number 4, you had to give me enough information to get 2 points. So a couple things I was looking for example, number 1, CDKs aren't activated until they bind with cyclin. When they are activated, they add phosphate groups to various target enzymes, and then that's going to then launch the cell into the next phase of the cell cycle. So that's ideally what I was looking for in those for that answer. Okay. It's a lot louder in there. And again, like I said, most of us did really well with that meiosis model. Last period before they did not. But we're just going to I gave you a blank one. We're just going to walk through this one more time and then we're going to see how this connects to what we're currently talking about. So humor me a little bit here. The cell we're starting with is 2N, which means that it is diploid. It has 2 of each chromosome. What does this second number tell me? That there's 2 chromosomes total, right? So it's diploid. There's 2 chromosomes total. By the end of interphase, what major events happened? Replication. When we replicate, we're making another sister chromatid, right? So we're going to go from having unreplicated single chromatids in our chromosomes to having replicated sister chromatids. Okay. And then on multiple quizzes, and I don't remember if it's this period or not, all of a sudden people had 2 chromosomes and then all of a sudden they had 4 chromosomes down here. So the replication has already happened when you get to this point. Okay, so you don't need to double the number of chromosomes. Okay, they're replicated at this point. Don't add a bunch of other chromosomes. So this cell is still 2N because it's still diploid. I have a homologous pair of chromosomes here, and I still only have a total of 2 whole chromosomes. So unreplicated is 2. When it's replicated, it's still considered to be just 2 chromosomes. You feel good about that? Okay. Between this and metaphase 1, what major event will have happened? Crossing over. If you read the directions really carefully, it told you specifically to cross over which one of the genes. Well, actually, it asked you to cross over the T. I didn't mark you off if you did the A instead, but specifically did ask you to change over the T. Okay. So we'll show that crossings over has happened by substituting that T allele for these chromosomes that are attached. Oh, sorry. Why did that? And the other important thing here is that these chromosomes are still paired up in metaphase 1. Okay, so they're not lining up individually, they're lining up as a pair. Okay, that is a mistake I saw in a lot of quizzes. So metaphase 1, they're lined up as a tetrad. At this point, we're still 2n equals 2. Between this cell and these cells, what's going to happen? Separation of homologous chromosomes. So if you're on this side of the plate, you're going to go to that cell. If you're on this side of the plate, you're going to go to that cell. Metaphase 2, we're going to be lined up in the middle here. And keeping track of my alleles. So this homologous chromosome has these alleles. This homologous chromosome has these alleles. What's happening between metaphase 2 and my final gametes? The splitting of the sister chromatids, right? So separation of sister chromatids. So this gamete is going to end up with dominant T, dominant A. This gamete is going to end up with recessive T and dominant A. This gamete is going to end up with dominant T and recessive A. And this is going to end up with recessive T. So how does this tie in to what we're doing now with dihybrid traits? Or dihybrid crosses? The alleles are getting shuffled randomly, right? You have the possibility of getting any combination of alleles, okay, between those two traits. Right? So even if they're on the same chromosome, we can still have independent assortment as long as they get crossed over. Right? So yesterday we said one exception to the law of independent assortment is if the genes are really, really close on the same chromosome, because in that case they're not going to get separated with crossing over, right? And they're going to end up in the same gamete. Right? So this is not going to go away. When we have our tests over this, you'll probably see something like this again. And when we get to the final exam at the end of the year, there's a chance you'll see it again. Okay, so this period, again, did much better than my other period, but okay, we just wanted to make sure we... Oh, so at this point, do I have two homologous chromosomes here? Well, I do, but they're in separate cells, right? So each cell now only has one of the homologous chromosomes. So I'm no longer diploid, I am haploid, and I only have one chromosome. So I got the impression that a lot of people are still really struggling knowing what those mean. So. Okay. All right. So if you can head back to your notes, I'm going to show you another method of finding genotype and phenotype ratios for dihybrid or further trihybrid, tetrahybrid crosses. So I appreciate if the phones go away. Yeah, it'll be at the end of the year. Yeah, but it will hopefully cover the cure up on the entire year. Okay, so please, please, please work along with me as we go through this. But one alternative to setting up a Punnett square is just using our laws of probability. Because if these are not linked together, I could find all my ratios by thinking about them separately and then multiplying them all together. Okay. So I could break this down instead of looking at both traits at the same time. I can think about what my genotype ratios are going to be for each individual trait and multiply them all by itself. So these are two heterozygous parents. What's the genotypic ratio always going to be for two heterozygous parents? 1 to 2 to 1. So 1 out of 4 is going to be homozygous dominant, and 2 out of 4 are going to be heterozygous, and 1 out of 4 is going to be homozygous recessive. Same thing for my other trait, since both parents are also heterozygous for that trait. 1 out of 4 offspring are going to be homozygous dominant, 2 out of 4 heterozygous, 1 out of 4 homozygous. So to find the genotype ratios, I have to multiply each one of these genotype probabilities by each one of these genotype probabilities. And we can do so pretty easily if we set up something called a forked line diagram. So here's how it works. I'm going to start by writing my first crosses possibilities in one column. So there's a quarter chance that the offspring will be homozygous dominant. And then I'm going to leave some space. There's a 2/4 chance it's going to be heterozygous. Then leave some space. 1/4 chance homozygous recessive. I need to multiply this by every possible probability of the second trait. 1/4 homozygous dominant, 2/4 heterozygous, 1/4 homozygous recessive. And I'm going to repeat that for my other two probabilities. 1/4 homozygous dominant, 2/4 heterozygous, 1/4 homozygous. So when I multiply all those possible combinations, I'm going to multiply every genotypic probability for my Y cross and every genotypic probability for my R cross. So go ahead and do that. So multiply 1/4 times 1/4 is 1/16. Is going to be dominant Y, dominant R, homozygous Y, homozygous R. Okay, so go ahead and do that for each one of these. Might seem like a lot of writing, but you'll be glad if you have this example written down.
Oh,
Is it a quarter this? Yep. 1/4 times 2/4 is 2/16. You guys do know how to multiply fractions, right? Is that something you need to review? So you multiply the numerator and you multiply the denominator. So the reason personally that I prefer this over the Punnett square is then I have my phenotype ratio or my genotype ratio all here instead of having to go back and like, oh, how many squares have this genotype and how many squares have this genotype. So I think as long as you take the time to set it up and you understand why it's set up this way, this is a lot faster and a lot easier and a lot more accurate. Because people make mistakes all they like can't count how many genotypes they like the squares are like I can't find everything. Anyway, so that's I think the benefit and kind of beauty of this. And of course, if you have the genotypes, then you can find the phenotype ratios, right? So how many are going to have both dominant traits? You have this one, and this one, and this one, and this one. So 1 plus 2 plus 2 plus 4 is? Not 7? 9.
7.
And how many are going to have a dominant Y and recessive R's? 3. And how many are going to have recessive Y's but a dominant R? 3. And then, so you get a 9 to 3 to 3 to 1 phenotypic ratio that we're expecting to find, right? When you do two heterozygous cross for 2 traits. This gets even simpler if the question doesn't require you to find the genotypic ratios. If you just want to find the phenotypic ratios, much smaller diagram. Because what's your phenotypic ratio from this cross? 3 to 1. Same for this. 3 to 1. So the diagram's a lot smaller. I have 3/4 that are going to have one dominant Y, 1/4 that are going to have recessive Ys. 3/4 that are going to have the dominant R trait, 1/4 that's going to have recessive R trait. Guess what ratio I get? 9 to 3 to 3 to 1. 9/16 that phenotype, both dominant. 3/16 this phenotype, dominant recessive. 3/16 recessive dominant. 1/16 all recessive. So again, I think this is, if you understand what's going on here, hopefully you do, we're just multiplying the probabilities together for each trait. I think it's a lot easier than setting up the Punnett squares. Questions? I'm going to give you one more similar cross. It's going to be a little bit more simpler. We'll see if you can figure out how to set it up. So, I do have this all keyed into the PowerPoint if you want to go back and look at it again sometime. So here's the cross I want you to try to set up in this way. So, think about what your ratios are going to be for the traits individually. Kind of compare with your group members when you're done. So if you think about these individual traits, now there's a 1-to-1 ratio of genotypes for offspring here, either homozygous dominant heterozygous, similar for the R trait, half heterozygous, half homozygous recessive. So when you set those up, you multiply each one times each one, you're going to end up with 4 different genotypes, 1-to-1 to 1-to-1, 4-to-4 to 4-to-4. Phenotypically then, what's your ratio going to be? Yeah, 1-to-1. So it's either going to be yellow and brown or yellow and wrinkled, right? Those are what those traits are. So both dominant, one dominant, one recessive. So just another tool to help you find genotype and phenotype ratios for dihybrid traits. So if you pull out your worksheet that I gave you yesterday, there are 4 problems on the back side that are all dihybrid crosses. And I'd like you to try to set up at least 2 of them with forked line diagrams, but if you are still feeling more comfortable with the Punnett square, then you can use Punnett square for some of them. Okay, so whichever method you feel like you understand better, that's the method you can use. All I care is if you end up getting the right answer. Yeah. Be very careful. Make sure you start with the right parent genotypes. Because if you don't start off with the right parent genotypes, everything's wrong.